and welcome to this special series from Karger Publishers and the POLG Foundation. I'm Will Mountford, and this is our third and final episode in a short series hearing from clinicians treating and patients living with mitochondrial disease. If you've not already seen parts 1 and 2, be sure to check them out now. We've covered how mitochondrial diseases happen, and we've heard what those eventual symptoms are like to live with. The question now is, what can be done about it? As of today, mitochondrial diseases have no cure. But science is changing that. With the right research questions in mind, the dedication of countless staff around the globe, and, crucially, the funding to pursue the treatments, technologies, and basic science underpinning POLG and many other diseases, there may yet be hope for patients and their loved ones. We go now to Professor Amy Goldstein and Professor Anu Wartiovaara to talk through the research and fundraising efforts pushing towards finding solutions for POLG. So, to start us off, and with going back to our previous interview with Jeff, the patient, he mentioned his willingness to throw himself onto every needle there was, to be part of any trial that there could be, to say that he's, an adult patient. Many patients don't make it to adulthood. He has his voice that he can use to raise awareness, whereas many young patients don't. So, how can one patient or one person affected by POLG give of themselves, and to how many trials?
It's a lot to go through, right? The trials themselves are exhausting. It is a lot of sampling. It's a lot of time. It's a lot of transportation. It's a lot of a burden, is what it is. And so I think that the patients that are willing to participate in these trials, many times they are compensated for their time and travel and participation. But the question is it enough? And is it taking a toll on them for that burden? So I think this is something that we worry about. And I would not want to stop anyone from participating in a clinical trial if they want to. I mean, trials are voluntary. We need we need patients that are willing to participate, who have the disease, to be willing to participate in these clinical trials. But I think we need to be very aware of the burden and the cost that it is on this patient population.
And there we need international collaboration, which is not totally easy because of different regulations between the states and the Europe, for example. But there's a lot that could be improved, especially in the possibility to collaborate worldwide.
Absolutely. And I think as much as we can, doing remote visits as opposed to in-hospital visits would improve the burden to the patients.
Now, clinical trials take people and they take money. So where can the most difference per dollar value be made?
We've seen a lot of grassroots type of involvement, right? There sometimes it's one family, right? When a family has a child with this diagnosis, I've seen amazing efforts happen on behalf of a child. And then we have partnered with organizations like the United Mitochondrial Disease Foundation, the UMDF, which has worldwide reach. But we just had our local walk, the Delaware Valley Walk, last weekend. And it's a wonderful opportunity for people from the hospital to meet with local families. And we gather at, the Navy Yard and have a walk together. At no effort is too little or too small. I have families that have done golf outings locally, and again, whether they raise $5,000 or more, every amount of money and awareness, I think, means something. And some of those families have been doing that for years, and it's very cumulative. And I do hope that we are saying something about the POLG Foundation and their efforts. And that's not small money and a small foundation at all. But I think that they've done an incredible effort to help raise awareness and money for POLG as a cause. And I think that hopefully will continue to grow over time as well.
Yeah, I think the Polgamma Foundation has been crucial, at least if I'm thinking now from the European view, to take the patients together. Polgamma disease is anyway a common disease within mitochondrial disorders. but the problem is that it's so variable. So if you think about the disorders, I've been thinking of this almost like the whole field of neurology that can be kind of a manifestation of Paul gamma disease. And that may be also something that thinking of large funding could be a strategy to apply, because you have Paul gamma can manifest as Parkinson's disease. Why does it cause Parkinson's disease? Is this also relevant for more common Parkinson's disease if we figure out Paul gamma for Parkinson's? Because these common disorders have very rarely this kind of a handle of genetic background as we have here. Why is it that the teenagers are prone to epilepsy? Why is this interesting for more common teenage onset epilepsy? Paul gamma disorders also, the patients have many kinds of they have depression, they have different kinds of psychiatric problems, which are not always occurring in all mitochondrial disorders. This is also something that we could understand the mechanisms through understanding Paul gamma disease. And when we think about kind of using rare disease as a tool to understand more common disease, that could bring in larger funding that would then benefit back to the Paul gamma patient and whether it would be through the foundation, I think it's a great organization to coordinate it.
Okay. Anu, could you tell me what it is going to be that turns that research into treatment? Is it just going to take time, money, people's attention, or all of the above?
I think we have to at least, again, looking at what we can do here, we should have built these kind of clinical trials units that can then have a very strict review process of different preclinical proposals that which has more evidence than the other. And then when you enter this unit, then you have the clinical staff, the nurses, the biobanks, the lawyers to actually get the ethical permits, the patient recruitment and everything. So these kind of units do exist in Europe, that they are very rare. So from my perspective, this is what I would need to actually take our findings then forward. And then also, if these kinds of units would be located in different countries, there's easier collaboration, and the output, I think that would be much more efficient.
And from the patient perspective, how are things changed in 2025 from, say, 2015, and where might we be in 2035?
Yes. So in the U.S. right now, we are having, not to be too pessimistic, but we're having a very bad year. The FDA has just rejected 2 drugs that were in front of them for mitochondrial disease, right? And so when I think about the future, in terms of running these clinical trials, I'm being extremely, I don't want to say critical, but my eye is on not only, how effective is this drug going to be, but how are we going to get the FDA to approve this drug, right? Because we need to be able to believe that this drug works, but we also need to make sure that the FDA is going to approve it. And so that takes me back to, what are the outcome measures that we are using, and how are we going to show statistical significance at the end of the day with this data? And it gets back to something Anu said earlier. Do we understand the natural history of this disease? And what that means for POLG is that we can't just do a single trial on everyone with POLG. We need to separate the young children with epilepsy from the adults with ataxia and neuropathy. Those are 2 different diseases. You cannot put them all in a single clinical trial. There may be different treatment modalities for those 2 separate groups. And, the mutations, the actual individual genotypes may be very, very unique here. And, there's been a very long debate about the genotype phenotype correlations and whether there is one or not for POLG. And I think that there is. And so, again, these things, we're still seeking to understand the natural history of this disease. And I think we're still lacking biomarkers for understanding progression in this disorder. And that makes it really difficult when we want a biomarker to really help us understand what has the progression been in this disorder, and is the drug making a difference for the disorder. So all of those things are very important when we're talking about drug development and treatment approval.
There's one very interesting development occurring, which completely arose from basic science, understanding how Pol gamma binds DNA, and there are these compounds now being developed to increase the interaction of Pol gamma, kind of the stability of the Pol gamma complex, and I think that the data look very interesting, and this is also going to drug development. This is not by us. But I think it's there. So that's why I said there's hope. There's different kinds of approaches being tested, and I'm sure that there's going to be some kind of help.
And what would you hope to be able to tell a patient in 2035, say? 10 years from now, someone comes in to the clinic and they've had the assays done, they've had the test, confirms they have a POLG mutation. What would you like to be able to tell them? And what is stopping you from being able to say that today?
Well, I have to say that there's been beautiful examples and surprises in disorders where I thought that there's not going to be therapy, at least if not gene therapy. One of them being the thymidine kinase 2 deficiency, TK2 deficiency, where there was a finding that nucleotides are depleted, and although the basic scientists who were studying nucleotide metabolism was saying that there's no way that oral supplementation will ever reach muscle, and still it does. The more efficient we are, the more collaboration there is, we will understand the basis of disease, because that's really the basis of the breakthroughs in these disorders. So I do think that example, also our examples of mitochondrial myopathy with this NAD treatment, which is not curing the disease, but it's delaying the progression, and sometimes even giving some skills back that were not there anymore. So those examples show that this kind of I called it metabolic bypass therapy, that those kind of those kind of understandings and findings can actually give hope, and their research is the key.
Yes, I hope that we have gene therapy for POLG in 10 years. I think that is a very realistic possibility at the pace that we're going right now for gene therapy. I have had many patients that have had the parents have had prenatal testing done. Their parents are undergoing IVF, and this is now part of IVF testing. This has been a prenatal diagnosis, so we're knowing early on, even before symptoms now. So I think that being able to treat early with gene therapy is a very high possibility once gene therapy is available. So that's one treatment. The other possibility that I'm really hoping for is that we are able to decide what we're going to do about the connection between POLG and immune, the immune connection here. We know there's an immune connection. There's an interferon signature. What are we going to do about this? We're not treating it. Some of us do try to treat it at times, or they're being treated before we know it's POLG with either steroids or IVIG. But there are other medications that are being used by immunology, rheumatology, that we are not accessing right now. And I think we are missing some treatment modalities because we just haven't figured out what is the right modality for our patients.
I'm always kind of saying that when we find it in somewhere, we have to say that this is where we find it, and if somebody else finds it too elsewhere, then we can more generalize. But at least this mutation is affecting the innate immune response, and which is then causing, in our view, the early epileptic onset of the teenagers who have this mutation. So if we can now understand that mechanism and push basically or support that pathway which is defective, then we can push back the onset of the disease.
If we could go Amy and then Anu, who should hear this? Either funders, patient families, anyone else? And what should they do?
Mitochondrial disease has tentacles that are going to reach out to these other diseases. So whether we're talking about the Michael J. Fox Foundation for Parkinson's disease, the Hereditary Neuropathy Foundations, the National Ataxia Foundation, I mean, if we're able to figure out something unique about Parkinson's disease or ataxia or neuropathy or epilepsy that helps us with those other conditions, in terms of treatment or pathogenesis, we're going to be able to help that disease as well. And we need to be sharing that information across all these boundaries. So I think that the message should be across all of that, especially in the field of neurology. This is a very heavy neurologic disorder. Predominantly, and then of course other organ systems, predominantly GI, liver, but again neurologically, this is something that I think we need to be sharing with neurologic societies as well.
I do think that there is hope because there is so much activity on this now on different fields. I would like this to be seen by pharma who are considering treatment trials, because this is not something that can be taken through in 3 to 4 years. There needs to be patience, and especially the deep phenotyping, so that you would compare similar patients to similar patients instead of having, for example, Parkinson's patients with Parkinson's versus patients with epilepsy just because they have a POL gamma mutation. So I think we really need to have these subgroups, as Amy was saying, and really to think that who are we testing and what's the preclinical data. So this would be needing longer term effort, but I think then the results would be good.
That's all for this last episode and for our POLG series. Be sure to revisit episodes 1 and 2 and head to the links provided in the episode description for more from the POLG Foundation and many more organizations dedicated to mitochondrial diseases. Thanks again to all of our guest experts and to you for watching along. Until next time, goodbye.